And thank you for the organizers for inviting me to give a speech here in the webinar. And thank you for Edward Peske for giving a nice introduction about lignin biosynthesis in his seminar a couple of days ago. So today I talk about lignin biosynthesis in Norway spruce. So first I remind you about the lignin and lignin biosynthesis, and then I have selected certain recent spruce studies that I will share with you. So I will talk about the effect of apoplastic redox state on lignin biosynthesis. I talk something about monolignol biosynthesis and our recent studies about monolignol transport. So as you know, Norway spruce is a common important forest tree in Europe, and its wood is used both for timber and for pulp and paper production. Okay, I will hide this one. So lignin is a phenolic polymer in the cell walls. Basically, it's in the cells having secondary cell walls like water conducting vessels and tracheids, and support giving fibers and stone cells. So lignin, at certain stages of development, lignin fills in all the spaces between the cell wall polysaccharides. So this excludes water from the cell wall. So this gives cell walls water impermeability and strength. And lignin is a good mechanical barrier against pathogens. So there are three main monolignols that are used for lignin biosynthesis. Parakumaryl alcohol that gives rise to hydroxyphenyl units in lignin, coniferyl alcohol that gives rise to guaiacyl units in lignin, and synapyl alcohol that gives rise to syringyl units in lignin. And the difference is the methoxylation states of the phenolic ring. And in lignin, there are several types of bonds between these monomers. And as Edward told, there are differences in composition of lignin in different plant groups. Gymnosperms, like Norway spruce, the lignin is mostly composed out of coniferyl alcohol, that is guaiacyl units, with small proportion of H units. In angiosperms, like Arabidopsis and poplar, the lignin is composed of G units and S units with small proportion of H units. And there is a difference in water conducting vessels. There is a predominance of G units, whereas in support-giving fibers, there is a predominance of S units in lignin. In grasses and cereals, there are G, S, and now the H unit content is higher than in other plant groups. In addition, there are some phenolic acids like parakumaric acid and ferulic acid attached to lignin. Lignin is making linkages to polysaccharides. These are called lignin carbohydrate complexes. So this is a cross-section of Norway spruce. So lignin is, now I'm talking about secondary xylem. It is wood in popular language. This is, on the right, there is a microscopical section stained with safranin Alcian blue. Safranin stains lignified cell walls red. So you can see that most of the cells in Norway spruce wood, they have lignified cell walls. These are tracheids, water conducting wood cells. Certain cells have blue cell walls, and these are rays. These are kind of for transport in the radial direction. Here is the cambium that locates under the bark in this trunk. And the cambium is are the dividing cells that give rise to xylem, the wood inwards, and phloem outwards. And you can see that in phloem, most of the cell walls are non-lignified. And the composition of the secondary cell wall in conifers, cellulose is making about 40%, lignin about 1/4. Different types of hemicelluloses composed about 30%, and extractives about 4% of the dry weight of the wood. And depending on the woody species, the lignin amount varies from 20 to 32 about. In our studies, we have utilized developing xylem from mature trees as one material. And here I have collaborated with Professor Kurt Fagerstedt group for several years. Every summer we have a trip to forest in southern Finland. There is a clonal forest owned by Luke. Is it Luke now? Finnish Forest Research Institute owned it earlier. And we fell a tree every year. And then we scrape out. The bark is easily detached at this time of the year, about mid-June, late June. And basically here is the cambium. Basically, I think the bark is detached from here. So the developing xylem is on top of the tree now, and we can use knives, and it's like slicing a cheese. We can easily remove the developing xylem and freeze it in liquid nitrogen and store it in -80 freezer. So every summer we do this. The other system I have utilized is extracellular lignin forming cell culture of Norway spruce. This cell culture originates from Professor Lisa Simola's laboratory. She was my supervisor of my PhD thesis. And this cell culture is from 1985, so it's about 36 years old. So this callus culture was initiated from the seed embryo of Norway spruce. And in nutrient medium, the cell started proliferation and formed a callus. So it's kind of non-differentiated cells. And about 3% of the cells differentiate to tracheids. About 3% differentiate to tracheids, having secondary cell wall. So most of the cells have non-lignified primary cell wall. And we maintain the culli on solid medium, because when we transfer the cells on liquid medium, after some days, let's say 5 to 15 days, the culture medium gets cloudy and it's extracellular lignin. And then the cells die the cells do not survive after the lignin is formed. That's why we maintain the culli on solid medium, and then for experiments we transfer them in the liquid conditions. And it is easy to remove the cells via filtrating them away through a cloth, and the lignin and medium goes through, and then you can centrifuge the lignin to the bottom of the centrifuge tube, and you can characterize it with NMR, and that has been characterized. And here is a table showing the linkage structure between the monolignol units in lignin. So as compared to artificial dehydrogenation polymer and mildwood lignin, that is lignin coming from the native trees, the beta-O4 linkage structures is about middle. The amount is about middle between these. Beta-5, it's more close to mildwood lignin content, whereas beta-beta linkage type is similar to that in the dehydrogenation polymers. This 5,5,0,4 structure, on the other hand, that's very similar to that in mildwood lignin. So we can say that spruce extracellular lignin is intermediate in structure between wood lignin and in vitro produced lignins. And that's why we think it's a good model to study lignin biosynthesis. It has also been shown that there are elevated amounts of ACE units as compared to wood lignin in this extracellular lignin. And it might be because these cells are kind of in artificial conditions, and they might be they are kind of in a kind of stressed conditions when they are in a shaking conditions, when they are cultivated in liquid conditions. Then there exist proteins and carbohydrates that are bound to lignin. And that was utilized in studies of LCCs, lignin carbohydrate linkages, that was conducted in collaboration with Martin Lavako. And we find out that there are benzyl ether type of linkages in extracellular lignin. Okay, now we come to the lignin biosynthesis. As Edward told, you need an oxidative enzyme to activate monolignol to a radical. So it can be a lactase, that is an oxidase, utilizing molecular oxygen, or it can be a peroxidase, utilizing hydrogen peroxide. And both types of oxidative enzymes, they make a phenolic radical, so there is a shortage of one electron. And when two radicals meet each other, a covalent linkage is formed, a dimer is formed, and when a dimer radical is meeting a monolignol radical, a trimer is formed, and so on, and eventually a polymeric lignin is formed. And when we started the studies of lignin biosynthesis in Norway spruce, of course, the idea was to characterize whether there is a specific lacase or specific peroxidase that is involved in lignin biosynthesis. But actually, there is a huge number of peroxidases and lacases in Norway spruce. The Norway spruce genome, the version 1 that is now available, it contains over 200 peroxidase gene models and over 100 lacase gene models. And many of them are present in the cell wall. So, or both of these enzyme types, they have broad substrate specificity. So that's why it's very difficult to characterize a specific enzyme, because many of them utilize, for example, coniferyl alcohol, the monolignol, in vitro enzymatic assays, but it's not a proof that they really work in lignin biosynthesis in plants. But that has been an interesting study. So our question was whether it's a peroxidase or lacase that activates monolignols for lignin biosynthesis. So we decided to scavenge the oxidant, hydrogen peroxide in this case. And by testing different agents to scavenge hydrogen peroxide, we find out that potassium iodide is the most effective. So we did the experiment where iodide treated cultures got 5 millimolar iodide at the beginning of liquid culture, and control cultures obtained a similar volume of water at the beginning of liquid culture. And here you can see that at day 5, the water-treated control cultures, they have formed a lot of extracellular lignin, shown here as a milky culture medium, whereas the iodide-treated cultures, after the same time of culturing, they are still clear. The medium is still clear, so there is no visible extracellular lignin. This can be quantitated. So in control cultures, there are certain milligrams of extracellular lignin per fresh weight of the cells after 5 days. In iodide-treated cultures, there is very little precipitate coming in these days. And basically, you can cultivate the cells up to 4 weeks in iodide-treated conditions, and extracellular lignin is not coming, basically. It's very little. We did a control with potassium chloride, the same 5 millimolar concentration added. And here you can see that extracellular lignin appeared. So it's not the potassium effect, it's the iodide effect. And iodide scavenges hydrogen peroxide. So this experiment shows that hydrogen peroxide is needed for lignin formation. So peroxidases have a role in lignin formation in the Norway spruce cell culture that we can conclude from this experiment. Of course, it's interesting to determine how much hydrogen peroxide exists there in the culture medium. Culture medium of these eucultures is basically a continuum of the apoplastic fluid that permeates the cell wall in native plants. It is just huge volume as compared to native plants. And in these experiments, I initiated small-scale liquid cultures, and then I measured the hydrogen peroxide levels in the culture medium by xylenol orange assay. It's an easy spectrophotometric assay. I can see that there exist micromolar levels of hydrogen peroxide, about 3 micromolar hydrogen peroxide level exists in the culture medium during lignin formation. I also added different substrates, putative substrates and inhibitors of enzymes that are putatively involved in hydrogen peroxide formation, and certain inhibitors gave indications about the enzymes that could be involved in hydrogen peroxide production in this cell culture, but I don't talk about that today. I will talk about what happens to monolignals when lignin formation is repressed by hydrogen peroxide removal. That is, we modify the apoplastic redox state by scavenging the oxidant away from the medium, or at least reducing its amount. And we studied how gene expression was affected. And for this, we conducted large-scale phenolic profiling. This was done in collaboration with Wout Boeren and Chris Morel. And this is the setup for the experiment. At time 0, the cells are transferred from solid medium to liquid cultures, and we made a big volume of liquid cultures that we aliquoted to different culturing flasks. This was to ensure that the cells are similar in each replicative cultures. Half of the cultures got iodide treatment and half the corresponding volume of water. And then we allowed the cultures cultivate and took samples at these time points. You can see that in control conditions, the extracellular lignin is visible after 10 days. And in this experiment, they were cultivated up to 20 days. The cells remained alive. Whereas in iodide-treated conditions, there was no extracellular lignin up to 20 points. Sorry. After 20 days, we changed the culture medium away from these iodide-treated cultures and washed the cells with fresh medium to be sure that the iodide was totally removed, and then put them back to normal culture medium. And the lignin formation came back, and we again took samples. So both cells and culture medium were taken. So basically, the lignin forming conditions are marked here in red color and the non-lignin forming conditions in blue color. The lignin was removed by centrifugation, and the small molecular weight compounds were analyzed by LCMS separately from the cells and medium, and the GCMS analysis was conducted to assay the other metabolites. And out of cells, we could see 505 compounds, phenolic compounds, and identify 71 compounds. So the identification is really difficult. Even we collaborated with a group that has expertise and good equipment for this kind of analysis. But what we saw was that there are glycosylated di and oligolignals inside the cells. There are flavonoids and proanthocyanidins, for example. So oxidative coupling can occur also inside the cells. In the medium, that is the apoplast representing the cell wall space, there existed 278 phenolic compounds. Out of those, 20 was identified. So there were di and oligolignals and glycosylated dilignals. And we could follow how these compounds accumulate in time. And we can see whether there are differences between the lignin forming conditions and non-lignin forming conditions. Also, the transcriptomic analysis was conducted. It was a different experiment, but the setup was similar. At time 0, the cells were transferred to medium. Half of them were treated with water and half of them with iodide. And samples was taken up to this point. Iodide was removed and cultures continued until the lignin came back. And RNA was isolated from the cells and RNA was sequenced. So basically I show here the pathway from the shikimate down to monolignals. So in lignin forming conditions, certain enzymes, genes encoding these enzymes were upregulated, whereas some of them were downregulated. And we had a look of the peroxidases and lactases, and some of them were upregulated and some downregulated. We also saw that there are changes in other metabolism, like in iodide-treated conditions, non-lignin forming conditions, the cell division seems to be activated, and that we saw also in the gene expression level. So basically the conclusion from this work was that apoplastic redox state not regulates only the phenolic metabolism, but all cellular metabolism. So something about monolignol biosynthesis. In angiosperm species Ginny and Arabidopsis, non-lignifying neighboring cells contribute to monolignol biosynthesis of developing trachary elements and xylem fibers. Edward Beske et al. Has shown this, and Rebecca Smith et al. From Lacey Samuel's group. There are nice papers about this. So our interest was to find out whether this is the case also in conifers such as Norway spruce. This work was conducted in collaboration with Kurt Fagerstedt. So to remind you about the structure of the spruce wood, that is secondary xylem. So this is the tangential section. Okay, the trunk is cut in this direction. So there exist two main cell types, there are tracheids, these long tubes that conduct water. And then there are rays, and rays are they are living parenchymal cells that have storage function and transport in this radial direction, for example. Okay, then there exist also resin ducts. Here is a resin duct, so there exist epithelial cells around these tracts. So question is whether the ray cells, these are the neighboring cells now for the tracheids, whether they contribute to monolignol biosynthesis in Norway spruce. So in Norway spruce, the gene transfer technology, that's possible, but it's very time consuming and laborious. So that's why we didn't go that way, but we decided to utilize the laser capture microdissection. Under the microscope, you can cut specific cells with this technology. So it was Olga Blockina who worked here. So she cut the rays separately and tracheid separately from developing xylem. RNA was extracted from these cell types and sequenced. So we could see that similar type of cellular metabolism was going on in developing tracheids and in developing ray cells based on their gene expression. Gene involved in response to various stimuli, phenylpropanoid, carbohydrate, protein, and amino acid metabolism were highly expressed in both cell types. And when we have a look of the phenylpropanoid synthesis from shikimate down to monolignols, these are the genes encoding the enzymes in those pathways. So expression of most of the genes encoding enzymes leading to monolignols were equally active in both developing ray cells and tracheids. Basically, in case the color is same in this column and this column, then the expression is same. If there is a cross, there is some difference. But basically, it suggests, the transcriptomic data suggests that at least the gene expression is up, leading to monolignol. So it suggests that the ray cells are probably contributing to monolignol biosynthesis. But this is not the final proof. So we wanted to do the metabolomic analysis to see whether there exist these metabolites, that is phenolic compounds involved in lignin biosynthesis in ray cells. And for this, we collaborated with Hiroshi Wada in Japan. So he has the equipment with whom you can inject cell sap from a single cell, and you can immediately analyze it with the MS. So it's called PPC MS analysis. So basically, we visited Hiroshi's lab with Olga Blockina in spring a couple of years ago, when the xylem development was initiated. And for this work, we bought seedlings of Norway spruce, because in the laboratory, you take the seedling and remove part of the bark and put it here in this machine. And by using a stereo microscope, Yuto Hatayakema, Hiroshi's postdoc, he was able to inject, this is a graphical view what happened. In developing xylem, he injected this capillar to ray cell and took out the content of this cell. And this machine was turning around, and then there was a MS mass spectrometry here. So the needle immediately injected the sample here, and the machine analyzed what are the metabolites in the cell sap. And similarly, the contents of the developing tracheids were analyzed by MS analysis. And from the results, we can see that both monolignols and their glycoconjugates are present in both cell types. So not only in developing tracheids, but also in developing ray cells. So this shows that the biosynthetic route to monolignols is active in both cell types. And we can conclude that also in conifers, like Norway spruce, the neighboring cells, here ray-parenchymal cells, contribute to monolignol biosynthesis used for lignification of developing tracheids cell walls. Then there is a third study I want to discuss with you. So that is monolignol transport. We know that monolignals are synthesized in the cytoplasm. It means inside the living part of the cell, whereas the lignin is synthesized in the cell wall, and there is a plasma membrane between these compartments. So how are the monolignals transported to the cell wall? This has been studied a long time, but it's still unclear. It is not yet clear whether what is the form of transport. Is it monolignol alcohol, or is it a glycosylated monolignol, or whether it can be a dimer, for example. Here is the coniferyl alcohol. When it's glycosylated, the glucose is here, and it's called coniferin. There are research data in favor of the glycoside. For example, in certain species, in gymnosperms and in some angiosperms, monolignol glucosides accumulate in the gambial tissues at the time of when the cell divisions start. There exist this kind of enzymes that make these conjugates, they exist, like UDP glucose coniferyl alcohol glycosyl transferases. They exist in the cytoplasm, and they are able to make, for example, coniferin out of coniferyl alcohol and glucose. And also enzymes that liberate monolignol from the conjugate exist, like coniferin beta-glucoside, glucosidase liberates monolignol alcohol from this conjugate in the cell wall. Those have been characterized. But then there are also indications against the glucoside idea. For example, these glucosides do not exist in all species. Okay, the way of transport is not yet clearly known. There has been ideas that it could be Golgi-mediated transport, similar to how hemicellulose or pectin are synthesized. They are synthesized in membranous structures called Golgi, and then there are vesicles delivering those in the plasma membrane, and then the vesicles fuse and liberate the contents. So this was suggested at a certain time, but Lacey Samuel's group showed that it's not probable in case of monolignals. Okay, and then there is the idea of specific transporter proteins, for example, ATP binding cassette transporters. For example, in Arabidopsis, there is the a protein that has been characterized as parakumaryl alcohol transporter, but it doesn't transport coniferyl or synapyl alcohol that are the main monolignol units. Then there is the idea of diffusion through plasma membrane, and basically it would be a free alcohol because the glucose is making the compound hydrophilic and it's not going through the membrane via diffusion in that case. So there are papers related to that. So we wanted to find out what is the situation, how is the situation in Norway spruce. And this is the work of Enni Väisänen, who is the PhD student of me and Kurt Fagerstedt. We share the supervision. And she did the transport assays. So she prepared the membrane vesicles from the developing xylem. She also prepared membrane vesicles from tissue cultured spruce cells. And for control material, she utilized tobacco BY2 cells. These are non-lignifying, they don't lignify. So these were kind of negative control. So she prepared the membrane vesicles. Basically all cellular membranes were involved in those vesicles. And then she had radio-labeled coniferin. And she basically measured how much radioactivity these vesicles take in. And this graph shows that the membrane vesicles prepared from developing xylem transport coniferin. Also, vesicles prepared from tissue cultured spruce cells, they transport coniferin. But what is interesting is also that also the non-lignifying BY2 cells of tobacco, they transport coniferin as well, and equally well than developing xylem vesicles. That is what Annie saw. Okay, Annie tested several phenolic compounds to find out what is the most likely substrate that this transporter is utilizing for transport. So basically she saw transport of coniferin, and that is the glucoside of coniferyl alcohol, and she also saw transport of parakumaryl alcohol glucoside. But none of the other compounds basically. So there was coniferyl alcohol, parakumaryl alcohol, so the alcoholic form was not transported in her assays. The dimeric forms like pinoresinol was not transported, or its glucoside was not transported. The other dimers like larisylresinol or isolarisyresinol were not transported. And also the beta for O4-erol basically not transported. And I forgot to say that this transport was ATP dependent. So there needed to be ATP in these transporter assays, and then you see the transport. Okay, and Annie saw that this parakumaryl alcohol glucoside transport was also present in tobacco BY2 cells that do not make lignin. So that makes us wonder whether this transporter is involved in lignification at all. It could be involved in general defense, for example. Okay, then Annie did several inhibitor experiments with these transporters. And basically these inhibitors that she utilized showed that actually it's not likely that it's a plasma membrane where the transporter is located, but it's more likely that it's a vacuolar membrane. This is the membranous compartment inside the cytoplasm. Basically, its vacuoles are for storage function, for example. So instead of plasma membrane, the transporter seems to locate in vacuolar membranes, and it seems to be, based on the inhibitor profiles, it seems to be either MATE or MFS type transporter. And these transporters are making secondary active transport, meaning that there is an electrochemical gradient, like certain ions are transported, making the gradient, and these transporters are utilizing that for transport. So in this work, we also did some membrane proteomics in collaboration with Gunnar Wingsle in WWSC and gene correlation analysis. This was to find out candidate transporters. And the membrane proteome work was conducted with developing xylem of Norway spruce, with lignin-forming cultured cells of Norway spruce, and then there was developing phloem of Norway spruce. This was kind of negative control because it is only the stone cells that exist. Some of them exist in phloem. They have some lignin, but the other ones, other cell types, they don't have. And by analyzing the data, Annie got some candidates here. Then there was this correlation analysis with a selected set of lignin biosynthesis genes, and 4 published data sets were utilized in this study. So there is this Congeny database in internet. So there are expression pattern of different libraries. Then these are done in by Swedish scientist Jokipiet-Lukkari et al. She has expression data in her paper. Okay, we have the tissue culture data of Laitinen et al. And then Blockina et al. Have the recent paper that I just talked about, have also gene expression data. So these were utilized in the correlation analysis. And we got candidate transporters from these. And the proteomic data candidates were compared to this correlation analysis candidates. And we got a list of candidate transporters. And basically they are MFS type and ABC type transporters. And now we are continuing this work with Olga and Kurt and trying to find out whether some of them is really involved in monolignol transport. So to conclude my talk, peroxidases are essential for lignin formation in the lignin-forming cell culture of Norway spruce. Further work is needed to characterize the specific oxidative enzymes involved in lignin formation. Apoplastic redox state regulates not only phenolic metabolism, but all cellular metabolism. Similarly to that in angiosperms, neighboring cells, here rake parenchymal cells, contribute in production of monolignals used for lignification in developing tracheids of Norway spruce. And monolignol glucosides, coniferin, and paracumaryl alcohol are transported. But however, the transport seems to be over a vacuolar membrane. So further work is needed to characterize the monolignol transporter. And I want to acknowledge, I have listed here the people who have contributed in these works that I have discussed here. And there are several collaborators. I have mentioned some of them by name, but there are also some others as well listed here. And the funding has come from mostly Academy of Finland. So please ask me some questions if you have some.